Okay, here's another video talking about enzymes, just some basics about enzymes to help you with your study for Bio 20. So enzymes are proteins. They're made of proteins. They're a catalyst. A catalyst is something that helps to speed up reactions. So if something speeds up a reaction, we can give it the name a catalyst. A catalyst could be something that's non-biological in nature, but if it's made of a protein, then we call it an enzyme. So they speed up reactions, but they only speed up one reaction. They don't speed up any given reaction. It has to match. The enzyme has something called an active site. So this part here that in this representative diagram is all bumpy, we would call the active site of the enzyme, and that will match just one, or in this case two, substrates. Substrates are the things that are being reacted upon. So the substrates might be things like lipids or proteins or carbohydrates or nucleic acids in the digestion unit. We are going to be working with hydrolyzing enzymes where we're breaking apart. So in the case of hydrolysis, we're going this way. We're starting with one big thing, like a big carbohydrate, and we're breaking it into smaller pieces. When that substrate matches up with the enzyme, the resulting structure is called the enzyme substrate complex. That just means the substrates and the enzyme are stuck together. And that is something that only matches particular molecules. So for each thing we're going to try to digest or break apart or hydrolyze, we're going to need a specific enzyme. You're going to learn a bunch of these names in Bio 20 in the digestive system unit. For lipids, there's just pancreatic lipase, but there's a special consideration here because these enzymes are all going to be water soluble. They're going to be soluble in polar materials, but lipids are nonpolar. So they need to be emulsified. They need to be broken into tiny little droplets so that the enzyme can get at the lipid molecules. For proteins, we have a bunch of proteins. We start this digestion in the stomach with pepsin, but there's also trypsin, chymotrypsin, erepsin, peptidases, and proteases. They all help to digest proteins and break them down into amino acids. Lipids help to break down, pancreatic lipase helps to break down lipids into triglycerides. For carbohydrates, we've got salivary amylase in the mouth. This is the only one that starts its chemical digestion, its enzymatic digestion in the mouth. And then pancreatic amylase and carbohydrates come along later in the small intestine. Pancreatic amylase is, of course, made by the pancreas. Carbohydrates get broken down into monosaccharides like glucose. For nucleic acids, they start out as DNA or RNA. They're broken down by nucleases. We're very vague here. Carbohydrases, nucleases, proteases, peptidases. These all mean there's a bunch of them and we're not making you memorize them. So that's all right. Enzymes are reusable, but they're not undestroyable. They are not without their limitations. Enzymes can be worn out. Enzymes can also be denatured, which I'll talk about in a minute. So for digestive enzymes, we'll start with something big. It doesn't have to have two pieces. It might have 200 pieces, but two pieces that will stick into the enzyme. They'll fit in there perfectly. Once they fit in there perfectly, the enzyme will do, if we look over here, a little bit of a bend to hold them in place. This bend is part of what's called the Induced Fit model, which is just a little hug that it gives it to hold it in place so that it's sure to have a really good connection with that entire molecule. Here we can see a water molecule coming in to help break that bond. Here we can see water molecule represented another way. This is hydrolysis. Hydro, talking about the water, and lysis, talking about the breaking. Once we've broken it into two pieces, those two pieces are now ejected, and the enzyme can do its job again. Enzymes can do their job remarkably fast, thousands of times per second. Something that might take days to do naturally could happen in just a tiny fraction of a second with an enzyme. The enzyme is ready to go again over and over and over as long as that substrate is available. But those enzymes can be broken. If an enzyme has a complex 3D shape, that shape is important, and if that shape gets twisted or bent or folded in some way that it shouldn't, usually by heat, by acids, or by bases. So acids and bases just mean here at the wrong pH. So an enzyme that's supposed to work in your mouth will not work in your stomach. Your stomach has an acidic pH. Or an enzyme that's supposed to work in your stomach won't work in your small intestine because, again, your stomach has an acidic pH. This denaturing, or sometimes we just say cooking, because it's the same thing that happens when you cook eggs or meat, which are proteins. That process is not reversible. When you cool down an egg, you don't get a raw egg. You just get a cold cooked egg. The protein has been denatured. And remember, enzymes are proteins. And lastly, for enzymes, there is the concept of inhibition, something that's stopping an enzyme from doing its job. So one thing that could stop it is the enzyme gets denatured. Of course, if you break something, it doesn't work. But two other ways are competitive and non-competitive inhibition. For competitive inhibition, that's where something kind of fits into that enzyme's active site. But it's the wrong thing, and the enzyme never gets to do its job, so it never ejects that molecule. And it's just plugged up, and it won't ever be useful again. For non-competitive inhibition, something goes somewhere else, attaches to some other part of that enzyme, some unexpected part. And what that does is changes the active site. So if it changes the active site so that the active site no longer fits the right substrate, the enzyme again cannot do its job. So enzymes are things that help to speed up reactions. We are focusing on hydrolyzing enzymes. There's lots of them to remember. You need to know who makes them, which organs in your body or accessory organs make them, what they work on, what their products are. Lots to memorize there. But they do hydrolysis. They work on that induced fit model. They can be damaged by heat or the wrong pH. And also they can be blocked from doing their job by competitive or non-competitive inhibition.